Today's topic is a small topic. It's not a very large topic. So we'll be talking about circumventricular organs. Uh, so in detail. Now again talking about blood-brain barrier. Dr. Paul Ehrlich was the one who discovered blood-brain barrier, and it says that after IV injection of tryptin. After IV injection of tryptin blue, most brain areas did not get stained. So these areas were inside the blood-brain barrier. Some areas of brain did get stained, and these structures were outside the blood-brain barrier. That is, they do not have blood-brain barrier. And the circumventricular organs are such organs who do not belong to this, who belong to the later part of the group. Now, talking about the microstructure of these barriers, the microstructure of this. So again, there's a tight junction called claudin-occludin between the endothelial cells. They form a continuous lipid bilayer. And there's continuous basement membrane outside. So as you can see, this is the continuous basement membrane outside the barrier. And there is astrocyte, podocyte, which form complete circle. So these are the astrocyte. So as you can see in the picture, so the red arrows, they basically indicate lack of permeability. There is no permeability, hence the blood. Again, this is the cross section of capillaries in the CNS where blood-brain barrier exists. The cap forms, which you can see. These are the endothelial cells inside. And of course, the black covering is the basement membrane, which makes it a complete circle. And these are the junctions, the tight junctions around the endothelial cells, which basically facilitate the fluid in and out. Now, the non-blood-brain barrier, as we say, so this is the highly vascularized structure. It's the fenestrations of the endothelial cells. There's a permeable basement membrane, and the astrocyte, podocyte do not form a complete circle. Versus the blood-brain barrier, which is a complete circle, this is, you can see, it's more like an egg. So it's not a complete circle. The cap form, as you can see, this is the foot process of astrocyte that do not form the complete ring. The permeable basement membrane, and the inside is the fenestrations of endothelial cells. So the black ones are endothelial cells, and the inside is the fenestrations. So this is the cross section of a capillary of CNS where blood-brain barrier appears to be absent. So again, you can note the fenestrations of the endothelial cells. So these are the fenestrations. So it is not connected as it was connected. So as you can see, this is tightly connected with each other. This is an incomplete connection. So it's not something which is connected tightly. Hence, it is more permeable than blood-brain barrier, and of course more vascularized. So the red arrows indicate permeability, and the exception is that subcommissural organ has no fenestrated capillaries. Now, what is its role? Its role kya hai inka? So the role of transites are basically the transites line the floor of the third ventricle of our brain. So body of transite is in contact with the CSF. The multiple foot processes extend towards adjacent capillaries, and it provides conduit between CSF and blood. The transites play important role in certain non-blood-brain barrier areas. So as you can see, the yellow one is the transite, and they connect the capillaries to the third ventricle. Otherwise, the vascular structure will get disrupted. So this is the body of the transite, and these are the foot processes. So more like a hook to the capillaries, right? So they basically provide a conduit between CSF and blood, so that they can be connected to each other. Now, talking about circumventricular organs, so again, most non-BPP areas are around third ventricle and one in fourth ventricle. So they are they are called circumventricular organs. They are bathed by CSF and are also in contact with capillaries, as we saw here. The median eminence of tuber cinereum, the organum vasculosum of laminar. So these are the segmental organs. So it's median eminence of tuber cinereum, organum vasculosum of laminar terminalis, subfonical organ, the pineal gland, subcommissural organ, area postrema, wall of optic recess, and posterior lobe of pituitary. So these are various circumventricular organs. Now, what are their functions? So there are various functions. First of all, is the chemosensitive organ, then allowing sampling of blood, endocrine feedback and regulation, homeostasis, metabolic regulation, fluid electrolyte balance, detect toxins in blood, and CSF circulation. That is something which is questionable. So some studies say they do, and some studies say they don't. Now we'll take up each one of them individually. Now talking about median eminence of tuber cinereum, that's METEC1. So the tan sites, as you can see, these are tan sites. So tan sites line the floor of third ventricle and the METC. It has arcuate nucleus that detects endocrine feedback to hypothalamus via the transites. It releases the RH and IH. These are the hormones basically. So PIH, and maintains basal levels of hormones FSH and LH. So this you can see, it's the first floor of third ventricle. It's raised central part of the convex gray matter in ventral most part of hypothalamus. So the part is raised, and that's again the convex gray matter in ventral most part of hypothalamus. The second one is organum vasculosum of lamina terminalis. It has osteoreceptors. It regulates ADH release by supraoptic nucleus and provides vascular outlet for LH-RH by preoptic and GHIH by suprachiasmatic. Both are adjacent to lamina terminalis. So as you can see, they are basically it provides the vascular outlet of the LH receptor hormone, and then by supraoptic, by preoptic, and growth hormone by suprachiasmatic. Both are adjacent to lamina terminalis. It is also called as vascular organ of lamina terminalis. Now talking about subfonical organ. So this is on the inferior surface of fornix at level of foramen of Monro. So again, this is the third one. So this is the organ. You can see this is the, I don't know if they find. So this is the foramen of Monro, and so angiotensin 2 receptors, and of course the neurons from CFO, that's a suboptimal organ, project to SO nucleus and OVLT. So both are osmoreceptors, and fluid. They basically help in fluid electrolyte balance. So as you can see, the new one. So the if you could check the flow chart. So as you can see, it's a sympathetic stimulation, hypotension, and decreased sodium in the DCT macula densa, which ultimately triggers the kidney to release renin. The angiotensin and again angiotensinogen, which releases A1 and A2, the ACE receptors in the lungs. basically causes cardiac and vascular hypertrophy, systemic vasoconstriction, increased blood volume. Just give me a moment. Sorry. Repeating again. So again, the sympathetic stimulation causes hypotension and decreased sodium from DCT macula densa. It triggers the kidney in basically the Ig cells and for release of renin. The angiotensinogen, along with angiotensin, also basically it releases the A1 and then A2, which is also triggers the ACE receptors in lungs. So A2 causes cardiac and vascular hypertrophy, systemic vasoconstriction, the thirst, causing increased blood volume. Again, the renin, sodium, and fluid retention, which again causes increased blood volume. A2 also triggers the nucleus, the SO nucleus, the suboxyphonical nucleus, causing release of ADH, and again leading to renal and sodium fluid retention. It also triggers the adrenal cortex to release of aldosterone, and again triggering the renal sodium and fluid retention, which ultimately leads to increased blood volume. So this is the cycle which basically is triggered and managed by the SFO. Now talking about the pineal gland, so the pinealocytes, that's also is also called as epiphyseal cells. So these are the this is the pineal gland, the fourth one, and again the cells are basically called as pinealocytes. Sorry. Now it's highly vascular structure. The fenestrated capillaries, it has fenestrated capillaries. It causes release of melatonin in the third ventricle. The melatonin is basically transported by transites in the floor of third ventricle and act on pituitary gland. So and if it acts on pituitary gland, so that it will cause release of hormones from the pituitary hormones. So as you can understand, the transites are basically the connection which ultimately helps in the transport of fluid, or probably transport of various substances, including hormones, from capillaries to third ventricle. So as you can see, here it's basically the melatonin which is transported by transites on the floor of third ventricle. So now talking about subcommissural organ. So this is a special epithelial, or it consists of special epithelial and glial cells. It has non-fenestrated capillaries. Functions are to circulate, in circulation of CSF in the hydrocephalus, and of course clearance of certain compounds. So it's below posterior commissure at third ventricle at the aqueduct of Sylvis junction. Now talking about area postrema. So area postrema it's basically it has modified